Hello and welcome to Unit 1, Lecture 3. Today we will be discussing general digestion, absorption, and the regulation of both digestion and absorption. First, there are two different types of digestion that occurs within the gastrointestinal tract. The first type is microbial digestion, and this is referred to as alloenzymatic digestion also. And basically this occurs within or outside the microbial cell in pregastric fermentation or in the lumen of the GI tract or in the large intestine. Then we have what we refer to as our host animal digestion, and this is autoenzymatic. Auto meaning self. And it occurs through a combination of denaturation and enzymatic digestion. It occurs within the lumen of the intestine. It occurs within the enterocyte, and it also involves hydrolysis. So first we're going to discuss the host animal digestion. So just in the last lecture where we discussed salivary glands and some of the types of saliva, here we're going to discuss what saliva is and its roles. So saliva is lubricating and hydration agent, and that is its main role. If you've ever eaten a significant amount of pretzels or crackers and you don't drink water, you definitely, if you have a mouthful of those, understand the benefit of having adequate saliva. It's more difficult going down if you don't have adequate saliva. So the main role of saliva is its lubricating role within the GI tract. Another major role is the buffering agent. We have a lot of hydrochloric acid that is formed within the stomach, so we need to buffer that so we maintain proper pH within the stomach or the rumen of the animal. We also have a minor role which includes starch and lipid digestion. So much starch and lipids can they start the digestion process. There are enzymes that are released from the von Ebner glands, and these are the glands beneath the tongue, and they secrete amylase to break down starch and gas and lingual lipase that starts breaking down the lipids. Now, it's a minor role because the overall amount of starch and lipid digestion that occurs is small compared to the amount that occurs within the intestine itself. It also is important for regulation of some endocrine hormones. So this includes epidermal growth factor, and this is actually signaling, it's a survival signal to keep the endothelial cells throughout the epithelial cells throughout the small intestine and the large intestine. It signals them to keep them alive. And then we have PYY. We'll be discussing this later in great detail, but PYY is mainly secreted lower in the GI tract, but there is about 1 to 10% that is secreted from within the saliva, and that helps regulate appetite. So depending on the animal, we have different amounts of saliva that is produced, different amounts of gastric juices, pancreatic juices, and bile that's secreted. So you could see here most significantly from the parotid salivary gland, the horse secretes about 10 liters per day, and this is a 100-kilo horse compared to that of a sheep that weighs between 30 and 40 kilograms. That's secreting 4 to 8 liters per day, and most significantly, the steer is producing the most at 40 liters per day. And this makes sense, especially given the size of the steer, the size of the rumen. That animal needs a lot more of the bicarbonate that's secreted. And remember that the parotid gland is secreting the serous saliva, which is mainly electrolytes and water, which is buffering the rumens of the sheep and the steer. We don't have the amounts of gastric juice that the horse and the steer are secreting. That's not to say that they are not. The sheep secretes about 5 to 6 liters per day. And then compared to the amount of pancreatic juice that's secreted, the horse has a lot more compared to the steer, especially if you correct it for the weight of the animal. And then the horse has 4 liters per day of bile that's secreted. Interestingly, horses do not have a gallbladder. We'll talk about the gallbladder in a little bit. So all this bile is coming directly from the liver. Sheep has significantly less bile than the horse. So the main thing that I want you to take away from this slide is that the steer significantly makes and secretes a lot more of the saliva. Horse has a lot more of the pancreatic juice. So once we enter the stomach, what's the important cells involved in initiating digestion? And this is all mainly due through chemical and enzymatic digestion. One of the first cell types that we encounter is the chief cells, and these chief cells are the ones that are secreting pepsinogen and gastric lipase. So pepsinogen is what's going to first be utilized to start degrading protein, and gastric lipase, we have some that is released. We'll talk about lipase in much greater detail when we are discussing the lipid digestion. And gastric lipase is important for human infants, for milk fat digestion, and rabbits, horses, dogs, and pigs also secrete a pretty significant amount of gastric lipase. Pepsinogen is a zymogen. I believe we discussed that terminology in the last lecture. So pepsinogen is inactive and it gets activated within the stomach. The next set of cells types is the parietal cells. Parietal cells are secreting hydrochloric acid, or HCl, an intrinsic factor. So hydrochloric acid is needed to denature protein, and it also converts pepsinogen to pepsin. So hydrochloric acid is activating pepsinogen to pepsin. So when pepsinogen comes in contact with that low pH, that's going to activate it. It also secretes intrinsic factor, and this is a protein that's very important for the absorption of vitamin B12. So this is the main reason why hindgut fermenters that are dependent on microbes to synthesize B12 for them, why a lot of those animals actually end up practicing coprophagy, because they cannot absorb B12 without it being conjugated to intrinsic factor. There is little protein digestion that actually occurs within the stomach, and little fat or carbohydrate digestion. Now, protein digestion does start in the stomach, so the pepsin starts breaking down those tertiary quaternary structures, and we're getting some longer peptides that are occurring. So here's a schematic of gastric digestion. So here we have our chief cells within these crypts in the stomach, and we have our parietal cells. The chief cells are secreting pepsinogen. The parietal cells are secreting hydrochloric acid. I would like to note that they are not secreting specifically hydrochloric acid together. Parietal cells are secreting hydrogens and chloride, and within the lumen of the stomach it becomes hydrochloric acid. The cell would not be able to maintain its intracellular pH if it had hydrochloric acid within it. So we have pepsinogen, we have hydrochloric acid formed within the lumen of the stomach. That is cleaving and activating the pepsin. And once pepsin is activated, it can also have some auto activation that occurs. So pepsin can also start cleaving and activating more pepsinogen. And you can see here, once we have our pepsin activated, it is breaking down protein down to peptides. Next, we move into the pancreas. And the pancreas is referred to as a accessory organ, but without the pancreas, no digestion would occur. So the pancreas is rich in buffers. These buffers are needed to neutralize the acidic digestia of the chyme from the gastric pouch. Oh, I would also like to mention regarding the hydrochloric acid, another important thing, not only does it activate pepsinogen, but it is also utilized to reduce the pathogens within the stomach. So that is a greater protection and greatly reduces the amount of foodborne illness that occurs within animals. But back to the pancreas. So the hydrochloric acid within the stomach has a very low pH, so we utilize the pancreas to secrete some buffers that we can neutralize that acid, because that would not bode well for the intestines if we have a lot of acid coming through and staying very low. Important to note that ruminants have significantly less bicarbonate that is released. So that is mainly due to the fact that they don't have as much hydrochloric acid that's secreted within their stomach, because they utilize the microbes. They don't need as much of that hydrochloric acid, so they have less bicarbonate. The pancreas is also has many enzymes that are involved in starch, protein, lipid, and nucleic acid digesting enzymes. So many of these are in the inactive zymogen form. Others are actually able to released in their active form. And then depending on the species, some species actually have cellulases and chitinases that are secreted. So some species, including crustaceans such as crayfish, the longhorn beetle, because they're eating a lot of plant material that has significant cellulose within it, and they don't have as adequate microbial aid, those animals specifically are able to secrete cellulase, which is going to break down the cellulose into glucose. So next we have this table showing the difference between the composition of the pancreatic juice of ruminants or a sheep specifically versus a man. So here we have, most notably, I want you to pay attention to the bicarbonate. So as I mentioned, ruminants have significantly lower amounts of bicarb that is secreted from the pancreas. So here you could see our bicarb in the sheep is 15 to 30 milliequivalents per liter compared to that of a monogastric human, which has 85 milliequivalents per liter. So that's a significant difference. Main thing that I want you to notice, the majority of the other components are pretty similar. Because the sheep has less hydrochloric acid within the gastric juice, their pancreas is secreting a little more chloride compared to humans, but the rest of the stuff is pretty similar. So main take-home message from this slide, bicarbonate is 15 to 30 in the sheep and 85 for the man. So I want you to know those numbers in red and be able to explain why that is. So I've been utilizing this terminology Zymogen over the last few lectures, and the Zymogen is enzyme that is secreted in an inactive form. So we have, first we have some enzymes that are catalytically active. These include amylase and lipase. So amylase and lipase, they're secreted from the pancreas. They do not need to be activated. Those two are active. They don't need any other cleaving of a component on that enzyme. So amylase and lipase are active. Nothing needs to be done for them to work. However, many enzymes are secreted from the pancreas in the form of a zymogen. So this requires chemical modification. And the reason why the body does this is we need to prevent self-digestion. This is the majority of the enzymes that are in the form of zymogens. They are proteases, meaning that they are breaking down proteins. So if the cells utilize proteins for their functional responsibilities, and we want to make sure that these enzymes are not breaking down the proteins that the cell needs to function properly. So we evolved that we have inactive, and they get activated once they are within the lumen of the GI tract. So two of the main types of proteases, one we've already discussed, pepsin, which is released as pepsinogen, and then the other one, so this is this is secreted from the stomach, and then we have trypsin, which is secreted from the pancreas. It's secreted as trypsinogen, it becomes activated and becomes trypsin. So some of the major enzymes that we have, pancreatic amylase, this is required for starch digestion. It is not secreted as a zymogen, and it has limited activity by the pH in the ruminants. So we have optimal levels that each enzyme is able to work and utilize. So amylase is not at the optimal pH within the lumen of the GI tract of the ruminants. so therefore the activity is limited in them, and it makes sense because the majority of starch we're going to assume is going to be utilized by the microbes and converted into various volatile fatty acids. So there shouldn't be too much starch coming into the small intestine of a ruminant anyways. Next we have pancreatic lipase. This is involved in lipid digestion. It is also not secreted as a zymogen. Also, keep in mind as far as exams go, if I repeat things, it's important to take note of that. So it'll be important to know which enzymes are actually zymogens and not zymogens, and I want you to be able to tell me those by name. And then finally, our pancreatic proteases. These are involved in protein digestion. All of these are secreted as zymogens. They are activated by intestinal proteases. There's an enzyme, enterokinase. So enterokinase is actually activating these. So trypsin, chymotrypsin, and carboxypeptidase. And also we have endopeptidase. Trypsin is an endopeptidase. Chymotrypsin is an endopeptidase. Carboxypeptidase is exopeptidase. So if we utilize our Latin, endo means inside, where exo means outside, meaning that trypsin and chymotrypsin, being an endopeptidase, they are going inside the peptide and cleaving it. So they are making these long peptides shorter, so they're cutting them in half, where our carboxypeptidase is cleaving off on the ends of those peptides and resulting in single amino acids and a peptide. So trypsin and chymotrypsin their products are resulting in peptides, and carboxypeptidase, its products end up being single amino acids and peptides. We also have nucleases, and these are involved in nucleic acid digestion. So all cells have DNA and RNA, and we need to utilize those resources. So we have nucleases that are breaking down the DNA and RNA within the cells that we're digesting, so then we can absorb those free nucleic acids and we can utilize those to synthesize our own DNA and RNA. Within the intestine, we have our enzymes, there are enzymes that the intestinal cells are actually secreting also. These enzymes are structurally part of the microvilli, so they are attached to the microvilli. They're not secreted into the lumen, and these are on the enterocytes, or the intestinal epithelial cells themselves, and these are mainly going to be disaccharidases and peptidases. So our disaccharidases are going to be breaking down disaccharides, including sucrase, which is breaking down sucrose, lactase, which is breaking down lactose, maltase, which is breaking down maltose, and trehalase. So which is breaking down trehalose, and we'll talk about those more when we are getting into significant detail on our carbohydrate metabolism. And then we have peptidases, so our trypsin and chymotrypsin, they are going to be resulting in dipeptidase peptides. So we have these peptidases that are bound to the membrane of the epithelial cells, and these peptidases are going to be breaking down those peptides, those di and tripeptides, into single amino acids, and they are also bound. We also have some of these peptidases are intracellular, so we have some di and tripeptides that are able to move into the epithelial cells, but once they get inside the epithelial cells, these peptidases within the cell are able to break them down into the single amino acids. Okay, so that's general overview of our autoenzymatic reactions, and we're going to get into much greater detail throughout the semester. So don't feel cheated on knowing all the great details of all these enzymes. We will get there later on. Next, we're going to discuss microbial digestion within the GI tract. So the first question is, how do these microbes get within the GI tract? And a significant amount of them are actually able, are first colonized within when the animal is born or hatched. So there is a significant amount of microbes that are within the vaginal canal. You also have the microbes from the rectum that are able to move over and can get within the vaginal canal. So when an animal is born, they actually are able, and they're going through that birth canal, they can, they will have those microbes all over them. First thing babies end up doing is they put their hands in their mouth, and so we have colonization that occurs then. And a lot of research is currently being done on this initial colonization. A lot of research is looking into the microbiome, so the composition of the microbes that colonize the GI tract throughout animals' different lives and how that affects the health of an animal. And research has shown, very interestingly, that the one question, for instance, was regarding how quickly it takes for a human baby to have the GI the microbiome become more similar to an adult. So when a baby is first born, if they are born naturally going through the birth canal, their microbiome within their GI tract is going to more closely look like the microbes that were found within the birth canal. And any babies that are born via cesarean section, their microbiome more closely looks like the what's found in skin. And it takes about 27 months for the microbiome of the GI tract to look more similar to what the microbiome of an adult does. So it takes a significant amount of time for that to change. In addition, it's unclear the benefits of the vaginal birth canal, those microbes that live within there. Some doctors actually feel so, they believe that there's something unknown and very clear that we need to encourage more women to have vaginal births if they can, barring any emergency, life-threatening issue that would require a cesarean section, because more typically babies that are born from cesarean section, they are more apt to have some allergies and stuff. So there's something beneficial that the bacteria within the birth canal helps the animal out. Much of that is very unclear. And so how does the microbes end up changing over time? A lot of that has to do with our environment. So depending on the food that the animal is consuming, and there's microbes within the dirt. We also have microbes within the air that are aerosolized, so we're inhaling them. So there is a significant amount of environmental effects that end up altering the colonization within our GI tract. And then usually when we have mothers that are grooming the young, so with humans, mothers are typically kissing their babies. So what we're doing when we do utilize that, when we're kissing our babies, it comes from this, you love the baby so much, you just want to kiss it, but really from a evolution standpoint, that innate feeling may actually become from we want to transfer our bacteria. The bacteria within our mouth and within our GI tract are good, so we want to make sure our babies utilize that. So we have grooming, kissing. So many animals will end up licking their young, grooming, so they are transferring microbes, so those microbes can colonize the GI tract. There are also microbes found within the mother's milk. In addition, there's also microbes found within the amniotic fluid. So for decades, it was always assumed that the womb was sterile, that the fetus was developing in a sterile environment. There wasn't any microbial colonization that occurred until the animal was born and going through the birth canal. But research has shown that if you take the fetus at different times and you look for microbial colonization, you can find microbes that have already colonized the GI tract, and those microbes are coming from the amniotic fluid. So what are our sites of microbial digestion? First, we have our pregastric forestomach, or our pregastric fermenters. This is occurring in our foregut fermenters in the rumen, reticulum, and omasum, and our ruminant species. We also have that crop in the poultry, which is the pouch of the esophagus, and there is a couple poultry or avian species within that actually utilize pregastric fermentation. Most notably is the Hoatzin, which is found in South America. You can find Hoatzins, and they utilize the pregastric fermentation that occurs. And then there's also thought that the kakapo, the nocturnal parrot that's found in New Zealand, there's some research that shows that they may also practice some microbial fermentation that occurs within the crop. We do have some microbial digestion that can occur within the stomach, and that's mainly the lactobacillus that are producing lactic acid. And then in our small intestine, we have adequate amounts, or pretty significant amount of microbial digestion that occurs, and the amount of microbial digestion that occurs increases as the digesta is moving through the tract. So you're going to have more microbial digestion occurring in the ileum than you're going to have occurring in the duodenum. And then in the cecum and large intestine or colon, there's a very significant amount of microbial digestion that's occurring. So what are the type of microbes that we have? We have bacteria, which is probably what the majority of you think of when you're thinking about microorganisms. We have protozoa and fungi and some yeast and bacteriophages. And our microbial classifications, we have anaerobic. So these microbes cannot tolerate oxygen at all. They are deep within the GI tract. This includes bacteria, protozoa, and fungi. If they are exposed to oxygen, it will end up killing them. Next, we have aerobic. These microbes need oxygen, and they are found within the oral cavity and the anal cavity, so the two ends that are most likely to be exposed to oxygen. And then we have the third group is the facultative, and these are bacteria that can exist with or without oxygen, so they are able to quickly adapt and change to where if they are first in the mouth, they can then, when they are in the depths of the small intestine, can switch to where they will not die due to an absence of oxygen within the small intestine. And they're basically found all throughout the GI tract, mouth, rumen, and the rectum. And these also include bacteria, yeast, and protozoa. So we have this ruminal microenvironment. So the rumen wall is the stratum corneum, which we discussed in the last lecture. Those are the dead cells. So you have the facultative bacteria are actually able to scavenge oxygen from the blood. So a lot of facultative bacteria live there. And within the rumen fluid, we have a lot of these microcolonies. So they can form little consortiums and exchange metabolic products. So they're kind of like little communities of microbes that all band together. And then the final grouping is the adherent bacteria, and these are bacteria that are actually able to attach to the particulate matter within the enterocyte. So here we have a schematic of the three distinctive niches. So here you can see the facultative bacteria are the ones that are bound to the stratum corneum. So these are the dead cells. The facultative bacteria are able to bind to those, and they're scavenging oxygen. In addition, urea is able to diffuse into the rumen, and we have the microbes are actually able to break down that urea and break it down to its nitrogen components, and then utilize that nitrogen for to make microbial protein. Here we have a free-floating microcolony. So we have some microbes that are able to bind together, little community that are exchanging products. They have a very symbiotic relationship. And then we have bacteria attached to feed particles right there. Here's a picture of lactic acid found on a starch granule. So all these little white components are lactic acid bacteria bound to a starch molecule, so they are starting to break that down, and they will be producing the volatile fatty acids off of that. Here we have a protozoa species, and you can see all the cilia. So this is what's allowing that protozoa to move around. So we have much of the things that you learned back in high school biology. We have protozoa with cilia and many of the other components. That's how they're able to move around the rumen. And then it was very interesting. It wasn't until the 1970s that we discovered that there are fungi that live within the rumen. Before that, it was thought that it was mainly just bacteria and protozoa. But in 1975, they identified fungus that was found within the rumen of the sheep. These bacteria or fungi are highly cellulitic, so where many of the bacteria and protozoa are unable to degrade lignin, these fungi are actually able to degrade lignin, and that is a main component making up cell walls. So the more mature a plant gets, the more lignin it incorporates into the structure, reducing the amount of cellulose. So a lot of bark, for instance, is made up of lignin. It's a very complex molecule, and we'll talk about that much later on. But because we have a lot of cellulose that is intertwined within these lignin molecules, having a lot of lignin decreases the cellulose availability by the microbes. So utilizing these fungi that can break down this lignin, you actually end up increasing the degradation of cellulose by 26%. So that's 26% more efficient, more energy that animal is able to utilize. In addition, these fungi are able to penetrate the cuticle within the plant cells much better than other microorganisms can. They're able to penetrate this, and the other bacteria, etc., cannot cross that cuticle. And that's one of the other reasons why we increase our cellulose degradation by having this fungi. And this is ubiquitous among all ruminants, and most likely all four gut fermenters, that they have these fungi and utilize these fungi within their pregastric pouches to increase the degradation of the lignin and increasing our cellulose degradation. So many of you, one of the hot topics is probiotics or direct-fed microbials. I'm sure many of you have heard of these. That's one of the major ad campaigns for various yogurt companies such as Activia, that it keeps you regular, etc. So what are the benefits of direct-fed microbials? These are feeding microorganisms to an animal, so we're supplementing it in the diet. So the thought is that we're giving good bacteria or good microbes that are going to improve the health of the GI tract. So one of the thoughts is that it inhibits the colonization of microbial pathogens. And this term is competitive exclusion. So the theory is that these microbes, the bad bacteria or the pathogenic bacteria, are not able to adhere to the intestinal tract. So therefore, if the bad bacteria can't adhere because there's no room, since we have these probiotics or good bacteria, that they're competing for space. So those bad bacteria are just going to move on along the GI tract and eventually be excreted, causing no deleterious effects to the animal. No, it's not going to be able to adhere and start secreting the toxins that usually are the main cause of food sickness. They also produce antimicrobial or bacteriocins that are going to prevent these pathogenic bacteria from colonizing. So if you were to think of two forces battling each other, so these bacteria, these probiotics are making enzymes and secreting them that are going to keep any of what they view as the enemy or the bad bacteria from colonizing. And then some of them produce unfavorable metabolic products, including lactic acid, so they're going to be altering the pH that is unfavorable to the pathogenic bacteria. And then they also have been shown to produce some neuroendocrine chemicals that actually alter behavior. So this has been a more recent finding that microbes within the GI tract are secreting hormones that, so if they don't like who's trying to move into town, then they will secrete some acetylcholine that's going to stimulate passage of or the flow rate, so those bacteria are less likely to adhere.
So here we have a picture of the proposed mechanism of competitive exclusion, and you can see here on the left-hand side, this is a treatment of birds that were given supplemental probiotics within their diet, and you can see this little X here. Those are actually the probiotic bacteria which are blocking the crypts of Lebercon from any pathogenic bacteria. Where when you look at the control birds that were not provided any probiotics within the diet, you can see all these bacteria that were able to adhere and grow within the crypts. So the probiotic bacteria are able to inhibit the pathogenic bacteria from adhering and maintaining their life within the crypts. So what are the end products of fermentation in the gastrointestinal tract? The main one is volatile fatty acids. These are short-chain fatty acids, and the main three that we will focus on in this class are acetate, propionate, and butyrate. There are also the bacteria themselves. When the microorganisms die, they are actually a source of protein that we refer to as bacterial protein or microorganism protein. So as those bacteria die and they move down the gastrointestinal tract, they are broken down, and their protein is able to be broken down by our enzymes, and therefore the amino acids can be absorbed. In addition, they also are able to synthesize many of the water-soluble vitamins. So one of the main reasons why a lot of our foregut fermenters do not have to have water-soluble vitamins supplemented into their diet, as opposed to, let's say, the hindgut fermenters and the simple stomach, is because the microbes are synthesizing those water-soluble vitamins or B vitamins, so they do not have to have them provided in the diet. In addition, if you add them into the diet, those microbes may actually alter them, and so what necessarily goes into the diet of a foregut fermenter is not always what is utilized We also have some fat-soluble vitamins, namely vitamin K, that the microbes are actually able to synthesize and we can absorb within our ileum and large intestine. And of course, many of you are familiar with the gases that are produced. We have carbon dioxide, methane gas, and hydrogen. There's a lot of, many environmentalists are concerned about mainly beef cattle and dairy cattle production of methane gas and various other greenhouse gases because they do have significant amounts of these greenhouse gases that are formed. But interestingly, some researchers at UC Davis have shown that they really do not end up resulting in very much of the greenhouse gases, especially if you compare it to how much people are driving. So what are the purposes of microbial fermentations? Why have we developed this wonderful symbiotic relationship between the microbes and the host animal? One is because of those volatile fatty acids that they produce. We've evolved, and various animals have evolved and adapted to utilize those volatile fatty acids so they can be used as an energy source. In addition, that microbial protein, those amino acids are able to be absorbed. Many of them can cross the cecal wall, so even though it's past the small intestine, they can still be absorbed in the cecum or in the large intestine. And then any animals that are, a lot of the foregut fermenters, ruminants, etc., they end up getting a lot of their protein from these microbes. In fact, many ruminants, if you look at their dietary requirements, they will have a nitrogen requirement as opposed to a protein or specific amino acid requirement. And that's because those microbes are able to convert that nitrogen into protein, and then the ruminants are able to utilize that microbial protein to meet their protein needs. And many other animals practice coprophagy, so many of the obligate herbivore hindgut fermenters, including the rabbit, they are coprophagous, so that means that they're consume their feces in order to get the benefit of this microbial protein. So just many of the obligate herbivores that do practice coprophagy, they are specifically consuming the cecal contents, which is very high in microbial protein. And once again, similarly with the vitamins, the foregut fermenters and coprophagous animals are getting the benefit of the microbes producing these, or synthesizing these various water-soluble vitamins. And vitamin K and vitamin B12 are able to diffuse across the epithelium. I'd like to mention that B12 in some of the obligate herbivore animals is able to cross the epithelium. Typically, you need that intrinsic factor to be bound to B12 for it to be absorbed, but some animals have evolved to not need that. So, what are the effects of diet and microbial end products in the rumen and the GI tract? So, we have a typical volatile fatty acid profile that occurs within the rumen. So when you feed cattle, sheep, goats, any ruminants, the diet that they adapted and evolved to eat, mainly grasses, hay, etc., they're going to have this very prominent typical profile. The vast majority of the volatile fatty acids, the main one is going to be acetate here. So you can see it takes up a large portion of this pie graph. Next is propionate. So propionate is the next most abundant. Thirdly, butyrate. And then we have some other volatile fatty acids that are make up about 1%. So dairy cows that eat an adequate forage diet. So you have to have adequate fiber, carbohydrates, non-starch carbohydrates. We end up having this proportion of these volatile fatty acids. So acetate has about 20 to 30 moles, propionate 10 to 15 moles, and butyrate less than 6 moles. Very important. I would like you to know these values and what's typical. We usually want a nice molar ratio of acetate to propionate 3 to 1 in the rumen. So if you take a cannulated cow and you open up the cannula and you take a sample and analyze the volatile fatty acids, we want a 3 to 1 ratio acetate to propionate. That's going to make a healthy rumen. But what happens when we decrease the dietary fiber? When typically in the diets we have, or in many feedlots, we provide those animals very high concentrate diets. There's a lot of corn, lots of grains, lots of starch is the main carbohydrate, very little fiber. So what happens when that occurs? So that fiber actually alters the volatile fatty acid that's produced. So we get a decrease in the amount of acetate, an increase in the amount of propionate. We're going to decrease the methane gas that's produced, and we're going to decrease the B vitamin synthesis that occurs. More specifically, we're most concerned about B12. And in addition, you end up increasing the amount of butyrate that occurs. So by having these high concentrate diets, we alter that ratio. So our acetate is reduced, our propionate is greatly reduced, butyrate is slightly, or propionate is increased, and butyrate is slightly increased. When we have these high starch diets, you end up getting a decrease in the saliva, and that's very critical because that's going to alter the pH within the rumen. So if you remember, the saliva within the ruminants, it's mainly coming from the parotid salivary gland. It's the serous saliva that has a lot of water and electrolytes, and those electrolytes are buffering the rumen. So when we have changes and we have high concentrate diets, we're decreasing the saliva, therefore decreasing the amount of electrolytes or buffers that are going into the rumen, which is going to result in a decrease in the ruminal pH. In addition, you end up decreasing the amount of ruminal fluid that moves down the GI tract. So what are the products of the initial products and the end products of microbial digestion? So here you can see microbes are able to use many different types or portions of the plants, including the cellulose, fiber, hemicellulose, and pectin. All of those are different types of fiber, including fructans, and we have starch. All of these eventually end up being converted to glucose and eventually pyruvate. And remember that the environment within the rumen is anaerobic, so there is no oxygen. So this pyruvate is not going to be going down through any glycolysis. So the microbes end up taking this pyruvate, and we get the formation of acetate, butyrate, propionate. That propionate can be converted into lactic acid, and we also have the formation of methane gas. So the ruminal volatile fatty acids can essentially be produced by any of the feedstuffs that the animal is provided. So how does volatile fatty acids in the hindgut and the cecum and colon end up occurring? So in the hindgut, this occurs due to the digestion of indigestible food. So the animal is not able to utilize it, so the microbes get the leftovers in hindgut fermentation. So they typically end up utilizing the cellulose and beta-glucans that are left over. Remember, the majority of host animals are unable to synthesize or produce cellulase, which is going to break down cellulose. They are also a source of energy, and depending on the animal, this may be very significant. So your specific hindgut fermenters, rabbit and horse, elephants, this is where the majority of their energy is coming from. They're obligate herbivores, but remember how inefficient it is to be hindgut. So if you remember previously with the one slide, the horse and the rabbit only get about 30 to 40% of their energy from the volatile fatty acids in the hindgut. These volatile fatty acids also help decrease the growth of pathogens, like we had mentioned earlier. They are releasing antimicrobial factors that are going to inhibit the growth of those pathogens. In addition, they enhance the growth of healthy gut tissue. So for instance, butyrate is very good for the immune system. It increases apoptosis. It stimulates the cells to proliferate. And so not only butyrate is produced, but some of the other volatile fatty acids have similar effects. And then they also alter the hepatic metabolism of lipids. So one of the benefits of increasing the amount of green leafy vegetables within your diet is it will help you decrease your blood cholesterol, and you will improve your lipoprotein profile. So if you ever go to the doctor and have your lipid profile analyzed, they're going to tell you the total triglycerides, your total cholesterol, your LDL, and your HDL. So the lipoprotein profile, you want a lower LDL and a higher HDL. So increasing the fiber is going to change that to where you have high HDL and low LDL. So here's another picture of how fermentation is utilized. The products of fermentation are utilized within the cow. So you can see here we have cellulose, hemicellulose, and starch come in. They're all converted to glucose, and then that is broken down to butyrate, acetate, propionate. It's absorbed in the blood, goes to the liver. All of those volatile fatty acids are utilized within the animal. So we have some forming ketones, and we have for instance, the acetates able to go and affect the muscles. We have ketones and acetate and glucose and the adipocytes, or the fat. And then we have within the mammary gland, the glucose and ketones and acetate are all contributing to the amount of milk fat. You don't have to have this picture memorized. It's just a great schematic showing you how everything is utilized within the animal. So here we have our breakdown, as I had mentioned, of the energy requirements. So here we have the sheep. They are getting 70% of their energy is coming from the volatile fatty acids produced within the rumen, compared to 5% in the cecum and 8% in the hindgut. Where if you look at cattle, in this paper they found 63% of their energy is met from volatile fatty acids produced in the rumen. 9% is coming from the hindgut. Compare that to the rabbit. You have the cecum here in the rabbit, 12%, 30% in the total hindgut, and then 30% in the pony or horse is where their energy comes from. So you can tell by looking at this that those foregut fermenters are much better adapted to utilizing those volatile fatty acids to meet their energy requirement. And then you compare all these animals to the human. The human is, along with the rat, one of the least efficient at utilizing volatile fatty acids to meet the energy requirement. So out of this table, things that I want you to know is you don't have to have all of them. Just generally, I would like you to know that ruminants such as cattle and sheep, 60 to 70% of their energy requirements. Hindgut fermenters are about 30, and human is about 6 to 10. So nutrient absorption. How do we absorb these short chain volatile fatty acids, or how are they absorbed within the rumen? So here we have this schematic, and you can see that for every, so this AC minus is our charged short chain fatty acid or a volatile fatty acid. This AC is abbreviated for acetate. So you can see that acetate is able to passively diffuse into the cells. However, we need to maintain the acid-base balance within the animal, so we cannot just take a charged molecule, a negatively charged molecule out of the rumen without replacing it. So in exchange, we end up pushing out a bicarb out. In addition, we have if the acetate is not disassociated from the proton or hydrogen, it's also able to passively diffuse in. And that is the best way, and it can just passively go straight into the blood. In addition, when we allow the charged acetate or the charged volatile fatty acid to enter, we also need carbon dioxide to come in. We need that carbon dioxide because we need to have this acetate neutral when it enters the blood. So we utilize carbon dioxide and water. We get bicarbonate, put one out. That hydrogen is going to bind that acetate and enter the blood. So some of the facts, this is the key thing, take-home message from the last few slides. About 70 to 75% of the volatile fatty acids are absorbed from the rumen in the animal. And these volatile fatty acids are passively absorbed, so they do not need any energy for them to be absorbed. Their absorption actually is better at lower pH. So one of the reasons why you may have asked yourself, why do we put animals on the feedlot if that diet ends up harming the papillae within the rumen and it alters this volatile fatty acid? It just seems like so much of this is bad news. Why would we want to do that to the animal? And basically, when we do that, the absorption is better. Remember, with that high concentrate diet, we're going to have less saliva, so we're not going to be able to buffer the rumen like we do in a hay diet, and so we have a lower pH. So therefore, the animal is more efficient at absorbing the volatile fatty acids at that lower pH, so the energy is more available for them. In addition, we have those undisassociated acids where it has the hydrogen still attached to it. They are more readily absorbed or diffuse into the cell than if they are disassociated. And the absorption of these volatile fatty acids much greater in the grain-fed animals, and that's mainly due to the change in the pH. So that lower pH is increasing our absorption, and much of that has to do with the lack of buffers. And we'll get into this in much greater detail later in the semester. So what are some types of absorption that occur within the small intestine? We're first going to start off with carbohydrates. So with carbohydrates, we have active absorption, and this requires ATP. So it requires energy, and this usually is in the form of the sodium-potassium pump that you may have learned in biochemistry or just general biology. And we also have secondary active, and this is the proton pump. So this is usually working in conjunction with that sodium-potassium pump or other energy requiring transportation within the cell. So an example of the secondary active is SGLT1, and that stands for sodium glucose transporter 1. We also have passive, which is where the nutrient is able to just move right through that cell membrane, right into the cell. And then we also finally have facilitated, where it's not passively diffusing through, but there is a transporter. There's a protein that the nutrient has to go through. It's almost like a tunnel that has proper charging that it can go through. So here we have a schematic explaining the SGLT1. We have a sodium and a glucose. Those bind to the SGLT1, and then that causes the transporter to flip, and we get a release of glucose and sodium within the cell. So that is the secondary active, SGLT1. There's no energy required at this point in time. However, we need to maintain this gradient within the cell. So, therefore, we use the sodium-potassium pump. So we end up pumping out that sodium out, and we pump in potassium. And this is an energy requiring step. So we actually utilize an ATP to make this happen. And then our glucose, here on the back of the cell, we have GLUT2. So this is a transporter. Glucose is just able to leave the cell through GLUT2 and enter the blood. So more specifically, we're going to talk about the SGLT1. So here the blue dots are our sodium, red dots are our glucose. And this light blue area is the lumen, and here we have the cytoplasm of the cell. We have an enzyme maltase here that's going to be breaking down maltose into 2 glucoses. So we have 2 sodium molecules that first bind the SGLT1. I know this picture only shows 1, but believe me, it's 2 sodium molecules, and once they bind, then that opens up this binding site for the glucose molecule. And when you have both the sodium and the glucose bind the SGLT1, we have the SGLT1 transporter actually flips within the cell. So the binding of the sodium and the glucose causes a transformational change, and that causes the transporter to flip in, and it flips glucose and sodium into the cell. So that is how SGLT1 works. So what are the types of absorption needed for protein absorption? So we have active transport, and there are 4 amino acid transporters. We're not going to get into them at all. The only thing you need to know is that there are 4 amino acid transporters. We'll talk about those more when we get into proteins. And then we also have passive. So this occurs with some of the peptides. They're able to passively diffuse into the enterocyte, and that occurs within both the small and large intestine. And then we also have active peptide absorption. So these peptides have their own transporter that requires energy So it helps them get into the cell, and we also have them occur within the enterocyte. Now lipids. Lipids are slightly easier. So basically we need an emulsion that occurs with the bile. So here we have our fat, we mix it with the bile, and we get the formation of the micelles. And it was always thought that fat just always passively diffused. But more recent research has shown that there are actually some fatty acid transporters. Well, when you have the lipase break down the triglycerides, we get the formation of a monoacylglyceride, also known as a MAG, where there's one fatty acid conjugated to the glycerol molecule. Those monoacylglycerides are able to passively diffuse into the enterocyte. However, when we break down that triglyceride, we also get a release of two free fatty acids. And as those free fatty acids, since they're charged, they have a more difficult time migrating through that enterocyte. So they actually use a transporter. So the transporter here is fatty acid transport protein 4. You don't have to know that specifically, but know that the fatty acids utilize a transporter. The monoacylglycerides are able to passively diffuse. The rest of this you do not have to know yet. You'll learn it much later in the semester. So how do we regulate digestion and absorption? So we mainly utilize the enteric nervous system, and this is the neural control of the gastrointestinal tract, and it's actually older than the brain. So when a fetus first forms, one of the first things that develops is the neural tube. So the neural tube is the first thing, so it actually is older than the brain and the animal. And it's semi-autonomous portion of the neuroendocrine regulatory system of the GI tract. So we can break this nervous system up into two different categories. And this is all autonomic, meaning that you have no conscious control over it. All of this occurs subconsciously. You're not able to control what your nerves are doing to your GI tract consciously. So the first one is the parasympathetic. And parasympathetic is mainly utilizing acetylcholine. That's the major neurotransmitter that it's using. And it's coming from the vagus nerve. So the vagus nerve is what's controlling the GI tract for the parasympathetic. The other category is the sympathetic, and its main neurotransmitter is norepinephrine. It has some other peptide neurotransmitter that it utilizes, and it doesn't have a specific nerve like the parasympathetic. You have many nerves that are controlling the sympathetic, and the sympathetic generally ends up decreasing GI tract activities where the parasympathetic is increasing. So if you have diarrhea, you can blame your parasympathetic. If you're constipated, you can blame your sympathetic. So here's some of the neurotransmitters, and we have this first portion, major neurotransmitters of the parasympathetic. This is acetylcholine that's stimulatory, and this is supposed to say sympathetic, not parasympathetic. I'm sorry, I thought I fixed that, but the sympathetic is generally inhibitory, and it is utilizing norepinephrine. So here, acetylcholine, it's affecting the nerves, muscles, secretory cells. It stimulates. Norepinephrine affects nerves, muscles, secretory cells, and it inhibits. You don't have to know the other neurotransmitters that are listed here. So what is the structural components of this enteric nervous system? So these can be split up into two different groups. We have the myenteric plexus and we have the submucosal plexus. So the myenteric plexus is clusters of nerves that are located between the muscles and the intestine, and the submucosal plexus is clusters of nerves located within the submucosa. And I like to show that the myenteric plexus, these clusters of nerves, are able to crosstalk between the sympathetic and the parasympathetic, which I'll show you in the next picture. So here we have a picture of the myenteric plexus and the submucosal plexus, so we can see how we have our parasympathetic here on the right, our sympathetic on the left. You can see this vagus nerve controlling the majority of the GI tract, where the sympathetic has many nerves that are controlling the GI tract. The parasympathetic upregulates the GI function, and they have receptors specific for acetylcholine. So here we have a picture of the myenteric plexus, and this is what I was referring to with this cross-talking that occurs. So you have the myenteric plexus with these nerve bundles, and you can see here the purple is the parasympathetic and the red is the sympathetic, and by them all sharing these little nerve bundles, they're able to crosstalk and control and throughout the GI tract. And here we have our submucosal plexus. We also have the sympathetic and the parasympathetic. Here you can see that we don't really have the crosstalk that's occurring. So how do we regulate the what's the endocrine regulation? What are the hormones that are involved? What is endocrine regulation? So for our endocrine system, we have three types of regulation. We have autocrine, and this is where the cell self-regulates. So it's releasing a hormone, and that hormone ends up affecting the cell. Next, we have paracrine, which is going to have a local effect. These cells are releasing the hormone, and it's going to affect the local cells around it. And then finally, we have endocrine. So these cells are releasing the hormone, they get into the bloodstream, and they have a systemic effect. So they end up affecting the whole body. So we have many different types of endocrine regulation that's occurring within the GI tract. So the main hormones that I want you to memorize or understand, there are 5 of them. The first one is gastrin. So gastrin is what's responsible for stimulating gastric acid secretion, and it's stimulated by the presence of protein within the stomach. So for instance, if you end up eating pure sugar, you're not going to have very much gastrin that's released. But if you eat a Snickers bar, you're going to have more gastrin because you have some protein found within that candy bar. Next, we have secretin, and this stimulates water and electrolyte secretion from the pancreas. So this is what's causing the bicarb to be released that's able to neutralize the pH. It's secreted by the S cells within the crypts of Leber-Kahn, and it also ends up inhibiting gastric acid secretion. So because it's controlling the pH within the duodenum, if the pH is really low, we want to decrease or inhibit the gastrin from causing any more hydrochloric acid to be secreted. So secretin will actually inhibit gastrin, and it stimulates the release of bicarbonate from the pancreas. Our next type of hormone is ghrelin, and this is secreted by the stomach, and it increases appetite. So this is all what's causing your stomach to growl is ghrelin. So when you have nothing in your stomach and you don't have those rugae stretched out, you're going to have the release of ghrelin. And so an empty stomach increases the amount of ghrelin. When you have a big giant meal, then that's going to inhibit ghrelin to hopefully signal you to stop eating. Next, we have cholecystokinin, or CCK, and this stimulates the secretion of pancreatic fluid and the contraction of the gallbladder. It does signal satiety, meaning that it's going to inhibit your appetite. So hopefully it will cause you to stop eating. If you're listening to it, it's released by the eye cells within the mucosal epithelium of the duodenum and the jejunum. And when you have a lot of fat and protein within your diet, that's what's going to cause its release. Makes sense, because we needed to cause the gallbladder to be released, the pancreatic enzymes to be released. We need the gallbladder, the bile from the gallbladder to absorb the fat. And a lot of the pancreatic juice is going to have proteases, which we need for protein digestion. And the final one I want you to know is peptide YY, and this regulates the flow of digesta throughout your intestine, and it enhances intestinal absorption. So I mentioned earlier that there's a little bit of PYY that's secreted in the saliva. Only about 1 to 10% of your PYY is found within the saliva. The majority of it is actually coming from your ileum and colon. And it ends up inhibiting gastric mobility and your pancreatic secretion. So basically, if you get down into your lower ileum and your colon and your cells are picking up that there's still a lot of protein and fat and carbohydrates, that's indicating that you need to slow down the flow of the digesta so we can be more efficient and absorb more of the nutrients, give it more of the enzymes more time to break down those nutrients and increase the likelihood of them being absorbed and increasing efficiency. It also increases water and electrolyte absorption within the ileum and the colon, and it helps reduce your appetite, which makes sense since it's mainly going to be signaled from the lower GI. If you have a lot of nutrients that are not being absorbed, we need to stop eating. So it's gonna help signal you to stop eating. So here we have a little schematic. We're missing ghrelin and PYY, but we have the other ones here. So imagine first that the stomach was empty, so our ghrelin would have been released, signaling you to eat. So when you signals you to eat, you go and eat some eggs and bacon in the morning. So a lot of protein. That protein's going to enter your stomach. It's going to signal gastrin. Gastrin's going to activate the chief cells to secrete pepsinogen and the parietal cells to release hydrochloric acid. Hydrochloric acid is going to activate the pepsinogen to pepsin, so now we can start protein digestion. As we move out of the stomach and the chyme enters the small intestine, we're going to have our CCK be signaled from those amino acids and the lipid involved, and we're going to also have the secretin that is signaled to be released. The CCK is going to have its effect on the gallbladder. The gallbladder is going to release the bile so you can better absorb your lipids. It's also going to signal the pancreas to release the enzymes. And these are coming from the acinar cells. And you're secretin going to your pancreas, causing them to release the bicarbonate into the small intestine. So it's not an all or nothing. All of these hormones and endocrine cells within the GI tract, they are basically dispersed in a gradient form. So here you can see gastrin mainly in the stomach, but you have some of it that's still released all the way down into the jejunum. Similarly, CCK, initially a lot of it is secreted in the duodenum, but it can go all the way down, and you can find some cells secreting it in the distal ileum. Similarly with secretin. Now, you by no means have to know this picture at all, but I think it's demonstrates really nicely how everything is released as a gradient. So our enteroendocrine cells, they are, as you just saw in the previous picture, they're dispersed throughout the GI tract. You can find them mostly in the crypts of the cells, and they have this flask shape. So they have a very narrow neck with the microvilli that's protruding into the lumen, and a very fat portion that is going towards the blood supply. And you have the frequency of the gradient exists for each cell type, producing the regulatory hormones and peptides within the GI tract. So all of them are utilizing a gradient, and all of those gradients differ depending on the main hormone that they are secreting. So here's a schematic of what the endocrine cell looks like. You could see here where I can say that it looks like a flask. So we have this narrow neck with these microvilli up here, this fat bottom, and you can see that the secretory granules, all these little black dots, all these little vesicles within the schematic, those are the secretory granules, and they're found throughout the cell. So they're not only at the basolateral side or at the luminal side, they're found all throughout the cell. And here we have the histological picture that's occurring, and the main take-home message from here, so this is from the small intestine, and you can see all these enterocytes, and basically there's a very low density of the enteroendocrine cells within the GI tract, especially when you compare it to enterocytes and goblet cells. There aren't nearly as many endocrine cells throughout the GI tract, but they are very effective, and they really don't need that much hormone to cause the signal that they need. Only a very small amount is required. So therefore, you don't need very many of these cells. And that's the end of the last lecture in Unit 1. And if you have any questions, feel free to contact me, and I would be more than happy to clarify any of the information in the following lecture.